In this video, we're going to discuss about the RNA modification. After the process of transcription, the mRNA is in the form of pre-mRNA. Okay, so it has to be modified. As I said, now this is your nucleus. So there is a mRNA. This is 5 prime to 3 prime. But it has to be modified. Okay. Now let me give you one example. This is your 5 prime and this is your 3 prime. Okay. So this is said to be your exon and this one is said to be intron, I-N-T-R-O-N, intron. Okay. So this is exon, this is intron, and this is your exon. So this is your mRNA. Okay. In the real structure it will be like that. And these exons, when we say exon, exons are said to be expressed gene. Expressed gene. When I say expressed gene means it is going to, its nitrogenous base is going to code for amino acid. Just say that you have A, U, G, C, A, U, G. Okay. So 3 nitrogenous base, okay, will code for amino acid, and these are functional genes. Okay, functional. Now, whereas intron is about intervening genes. Okay, intervening genes. So this phenomena, this kind of phenomena, is you will find in the in eukaryotes, not in prokaryotes. So likewise, I say such modification, because of that, such modifications are not required, okay, in prokaryotes. So these are said to be intervening genes, and when I say intervening genes, these are non-functional. Non-functional. So these introns will be removed, okay. These introns will be removed and only the exons will bind. Okay, we need only exons and we have to remove the introns. Such process is said to be splicing. Now, how those modifications take place? Okay, there are 3 ways. The first is capping. Now, what is capping means? Capping means at this 5 prime end, okay, at this 5 prime end, you are going to add methyl guanosine triphosphate. Methyl guanosine triphosphate. Okay, so let me add. In this 5 prime, I have added MGPPP. This is said to be capping. Now, I'm just removing this one and I'm just examining this 3 prime as here. Okay. So, second is said to be tailing. Tailing means you're going to add adenine residue, like around 100 to 200. So we also call as poly-A-tail. It means that you're adding adenine, maybe 100, 200, okay, maybe 150, depending on the processes or requirement in the process. So tailing is done at 3 prime end. So it is at 3 prime end. So this is poly-A-tailed. Once these 2 processes has been done, the third is called splicing. Splicing means removal of introns. Okay, so splicing is done by a specialized protein. Okay, we call as spliceosome. Okay, and what is this spliceosome? Spliceosome is a combination of small nuclear RNA plus protein, and we call as SNR. Okay, S and R and P. What is SNRP? We call just as SNRP. Okay, small nuclear ribonucleoprotein. Okay, small nuclear ribonucleoprotein. So this spliceosome, now what will happen? So this is your, just say that this is your exon, and there is a exon. So this is your intron. Okay, usually in intron, in this position there will be a GU sequence, and here it will be a G sequences. Okay, at the center there will be A, the branch point. Okay, so this is your intron. Now the spliceosome will come to an action. Spliceosome is usually having U1, U2, U5, U4 plus U6. Okay, the first U1 will come and bind here. Okay, so I will make a structure like this. U1 will come and bind here. This is U1. U2 will come and bind here. Okay, now U3. Okay, and then we have U1, U2, U5, U4, U6. So U5 here and U4 plus U6. Okay, so these are all coming in, binding to the intron's position. So what will happen? It will form a spliceosome. Okay, the structure is said to be spliceosome now. It's said to be spliceosome. So in that way, they will cut the introns. They will remove the introns, and there will be only exons. So this is your exon, this is your exon, and this is your 5 prime. So here will be GMPPP. This is capping, and here will be poly A tail, and this will be 3 prime. So now this structure of mRNA, this is said to be mRNA, and it is mature RNA. So this process, this modification is very important. Okay, the first one is capping, second one is tailing, and the third one is splicing with the help of spliceosome proteins. Okay, now once the mRNA is synthesized, now this mRNA will come out to the cytoplasm. Okay, they will leave the nucleus and they will go to the cytoplasm, where the protein synthesis will take place. To understand the protein synthesis, in the next class we are going to discuss about the genetic the genetic code and the process of translation. Thank you.